Overall, interphase is when the cell grows in size, resulting in the doubling of organelles, cytoplasm, and proteins. During interphase, the cell's metabolic activity is high. There are 3 different stages of interphase: G1, S, and G2. In G1 and G2, the cell simply grows in size, along with the amount of cytoplasm, organelles, and proteins doubling. In the S, or synthesis phase, the DNA of the cell is duplicated. At the end of the S phase, there are 2 sister chromatids from each chromosome. The entire interphase process takes at least 90% of the total time for cell division. Inside the nucleus, the chromatin fibers become more folded and coiled. As a result of this, they form chromosomes. These chromosomes are 2 identical chromatids attached to one another at the centromere. At this point in the process, the mitotic spindle starts to form as microtubules emerge from the centrosomes. The 2 centrosomes move to opposite sides of the cell. The nuclear envelope disintegrates into fragments and ultimately disappears. Spindle microtubules interact with the chromosomes by pulling the chromosomes towards the center of the cell. The proteins inside of the spindle microtubules help to pull the chromosomes to the center of the cell. The spindle microtubules attach to kinetochores, which are protein structures on each pair of sister chromatids. Finally, some spindle microtubules attach to microtubules coming from the opposite side of the cell. The mitotic spindle is fully formed, with each centrosome at each opposite end of the cell acting as poles for the spindle. The chromosomes are now located in the center of the cell at the metaphase plate, which is an imaginary plane exactly in between each pole of the mitotic spindle. The 2 kinetochores at the waist of each pair of sister chromatids is attached to a spindle microtubule coming from opposite ends of the cell. The spindle microtubules pull at the kinetochores, which pull the sister chromatids apart. Once each sister chromatid is separate, it is considered a chromosome. There are now 2 complete sets of daughter chromosomes at each end of the cell. The 2 poles are moved farther apart, and as a result the cell becomes elongated. This phase is over when the chromosomes reach the 2 poles of the cell. Daughter nuclei form as nuclear envelopes form around the chromosomes. The mitotic spindle disappears and the chromatid fiber of each chromosome uncoils at the end of telophase. In animal cells, a cleavage furrow forms and pinches the cell into 2. The cell completely splits into 2 identical cells. Cytokinesis marks the end of the mitosis process.
Make me smile.